Good afternoon, everyone. We get started with the post-lunch session of the Physics Conclave 2025 second day. So we have a lineup of very interesting panel discussion sessions and also the poster and other interaction sessions, so do attend. In this particular panel discussion, we have a broad umbrella of physics of soft, living, and active matter. And I request my colleagues Apratim Chatterjee, Shiv Prasad Patil, Ushashi Roy, and Vijay Chikkadi, and myself to actually be part of this panel. We're going to give a very brief overview of the topics of interest across our research groups. And then we're going to get going with the panel discussion, followed by questions and interaction with the audience. So we'll get started with Apratim.
Yeah, so I'm doing chromosome physics and in particular physics of life. Now this typically biology, life and physics, at least to me, didn't come together. I left my biology after my class 10, so I'll spend a minute to explain what is physics of life. So what I do is essentially soft matter. And what is soft matter is the physics of interacting macromolecules, and it's statistical physics of interacting macromolecules. Now, when you apply it to the biological context, you call it physics of life, and this is a term which I myself learned just 3 years ago, and I've put it physics of life on my website just this last August. So we can talk more about it during the panel. So the broad questions of physics of life is basically due to interacting macromolecules, statistical physics, non-equilibrium statistical physics of macromolecule. How does structure, organization, dynamics that we see in a living cell, how does that emerge deterministically? Why deterministically? Because the cells have a lifetime, we have a lifetime, we grow in a certain way out of stochastic processes in living systems. So how does life emerge? We don't know. At least I don't have any clue. That's a very big question. I basically am focusing on a smaller targeted question at the moment. And basically I'm trying to figure out what is the underlying mechanism of evolving organization of chromosomes in the bacterial cell E. coli. So bacterial cell is a small cell, 2.5 microns in length, 1 micron in diameter, cylindrical. And the chromosome has some organization as observed in experiments and it keeps on evolving. And to our surprise, we figured out that the bacterial cell uses entropy, physics of entropy. So for me, chromosome is a long polymer chain object with a huge amount of entropy, and it uses entropy to organize its chromosome during its life cycle, and in particular it uses loops, internal loops, to control different kinds of organization at different stages. That's a strong statement to make. Have I got it validated by experiments? Yes, we started from experiments, and then we talked with biologists. They helped us, they believed us first of all, and then said, okay, can you check out this mechanism, that mechanism, that's what's going on. So where does physics come into bio? I said that this is the physics of interacting macromolecules. That's soft matter. That's what I have been doing during my postdocs and so on and so forth. And it is essentially, soft matter is nothing but the statistical physics of weakly interacting molecules. Order of KBT. KBT, thermal fluctuations, are extremely important. It keeps the systems liquid, fluid, change conformations, and so on and so forth. And what I'm doing currently is I'm learning about entropic interactions. So typically people write Hamiltonians to describe energies. I put in loops, or my group puts in tunes entropic interactions. And we are learning how to do that because we want to apply that for chromosomes of higher organisms, which will take some time because we first need to know the physics, and I'm using polymer physics. So in my future work, plan is to look at topologically modified polymers. Topologically modified polymers with internal loops is topologically modified polymers to explain DNA dynamics. In particularly, I'm going to look at various active processes like extrusion. Extrusion is where a loop is pulled by some protein molecule. The effect of loops, supercoiling, there is twisting happening. And once I understand the physics, I'm going to apply it to more and more complicated system chromosome of higher organisms. And I also do active matter hydrodynamics, which we can talk on a personal level later.
Thank you, Kapite. So, to give an overview of what we have been doing in my lab, and also to give you a gist of what happens when you bring electromagnetics and statistical physics, is of interest not only from an optics viewpoint, but also from a soft condensed matter viewpoint. So one of the central questions we have been trying to address in my lab is to look at what happens when you drive soft matter. In our case, it is an inanimate, lifeless soft matter. But you actually can drive these soft matter systems to mimic certain things which are kind of emulating some natural systems. For example, you can have large number of particles driven by external force, and that particular external force can create these objects and bring in very rich dynamics. And in there, the specific kind of dynamics what we have been addressing is the effect of confinement, particularly optical and optothermal confinement. So that brings in the thermodynamics aspect of it, and also to a certain extent the hydrodynamics aspect of it. And then we look at examples of systems which can emulate something like what you are seeing on the screen, where you can revolve objects in a chiral way, because there is an assigned direction to these kind of movements. And how do they propel? What are the reasons they propel? What is the driving mechanism? And these are questions not only of fundamental interest to understand how statistical mechanics and electromagnetics can actually be brought together, but this is also of very high interest to understand how to induce forces in various different environments down to the few micrometer, and sometimes even sub-micrometer, all the way down to few nanometers. And understanding the momentum transfer is of very high interest to me. So therefore, I have been addressing the questions related to optical forces, torques, momentum transfer processes, using light as the main driving force onto the soft matter system. Yesterday, I also mentioned about the structured light, which has a very rich characteristics by itself. Of course, we use a variety of methods to understand these kind of processes. So what is the fundamental kind of overarching questions you can address in these kind of areas? One of the most important aspect is to address how to induce force in complex systems. Okay, and this is a very interesting approach to take, because you can also now look at some hydrodynamic effects which are otherwise not feasible to be implied and applied in small constraints. The second point is that you also have some new electromagnetic effects. Case in point is something called as negative torque, where you have something moving in this particular direction where you have an assigned direction, and you can now have objects going in the ulta direction. And why is that happening? It's a very interesting question. And you can actually emulate that in our laboratory. As I speak, we also have some particular data which shows that these kind of negative torques can be implied. There can be lateral forces, and there can be very unconventional, non-conservative forces which you can apply using such systems. We use a variety of tools. In fact, tools become very important, as we will discuss in our panel discussion. And this is the overarching principle behind my group. I need to underline the one point is that we look at things from a soft photonics viewpoint. That's where the soft matter aspect and the optics aspects combine with each other. And there is a lot of things to explore in this direction. So I'll ask Shiva.
Okay, so our lab is called Nanomechanics Lab, but before I go there, let me tell you the scheme of things here. In our lab, for a number of years, we were looking at liquids confined to scales which are of the order of nanometers. And this means that only a couple of molecular layers of water and other things. We stopped working on it about 5-6 years ago. And our current focus totally is on molecules which are responsible for life. As I said earlier, we use this tool called atomic force microscope, spring balance, let's say, which can measure forces of the order of piconewton. That's again 1,000 billion times smaller than 100-gram weight, as I said also earlier. This technique, the spring balance is being seen on the left-hand side. It's actually a cantilever with some probe attached to it, sometimes a spherical probe or a sharp tip. What we're trying to do here, so if you look at materials, typically our classification scheme of materials is solids and fluids, first order classification. Then we say fluids are liquids and gases. This started way back at time of Newton and Hooke's and Maxwell and so on and so forth. So these are like simplest classification scheme of material. The way to go about measuring or categorizing them is actually the simplest way is to press on them and see how they deform or attach a weight to it, see how they extend. And that's the area of mechanics, as nanomechanics in our lab. What we have done now is we are doing this at the scale of nanometers. So that's why we call ourselves nanomechanics lab. Things of interest, we've done organisms, cells, and molecules. I will not speak much about organisms. There we tried to look at how the spatial variation in elastic Young's modulus of a biological material of an organism actually helps it do locomotion by conserving energy and releasing it at times. And that was done some years ago. Currently, we are focused on cells and molecules. What we are doing on cells is that with a spring balance or a cantilever like that we showed there, we press on a single cell and see how it responds to this external force. And by such measurements, but we come to conclusion that the cells in general, and this is kind of consensus in the community also now, cells in general show soft glassy behavior. Why that comes about is a question that statistical physicists and non-equilibrium statistical physicists will have to give. But that's the current consensus in the community. And this is not only from statistical physics point of view is important, but what we have done in the lab is taken different types of cells, 10 or 15 different types of cells, and depending on what kind of disease the cell is acquired, we see these glass parameters of the cell distinct from each other, and this can become a very important diagnostic tool. The other thing that we do now, about 1,000 times smaller in scale, is we actually take a single molecule. On the screen you see is a globular protein. It's about 4 to 5 nanometers in size. What we do with AFM is pull on them, and as we push and pull on these molecules, we sort of, with certain force, here again 10 piconewton of forces, and the deformation produced in these molecules is of the order of picometers. Okay, so it's extremely small deformation. And for measuring such small deformations or extensions in the molecule, we use interferometer, which is very special and unique to my lab. Actually, no one else in the AFM community does this. And we have now the company is also interested in trying to see how this can be taken further. What we have learned from there is that the molecules are viscoelastic in nature. What that means is that it's not fully elastic, but if I make a cycle of pushing and pulling, then apart from being elastic response, it has a viscous loss. And this loss is quite interesting, and many statistical physics theories are saying this is entropy production and various ideas in, again, stochastic thermodynamics. But why is it relevant? Once again, if you look at these globular proteins, they are strung together in our bodies to deliver various functions. Particularly, suppose your heart pumps, blood, and the protein that's involved called titin, that's what we studied in the lab. This is a long protein, as again, this globular protein strung together. It sort of contracts and releases to have this function along with myosin and actin. And another example is that we have a tip link in the ear, which means, again, a cadherin molecules which are strung together, and they act like a rope to open a door, and the ion channel is open, and you see a calcium signal or electrical signal so that we hear things, and so on and so forth. And diseases related to all this hearing loss, cardiomyopathy in heart conditions, all of them are actually point mutations that are in these proteins. And we exactly don't know what these point mutations are doing, and the elastic measurements probably will provide a cue to this. So I will stop there.
I'll take over from where Shiva left. And for the paradigm of my research, I would like to invoke the multi-scale competency. It's a multi-scale, multi-component competency architecture in the living world. We need to remember that each of these levels, these are composed of interacting components, and they are solving problems in their own parameter space. And hence, they are interacting and solving problems. Some emergent behavior is coming up in the next level. And then these levels have also interactions. They have interactions, and there is an emergence of a emergent behavior is coming up. So I have taken some 3 illustrative examples. The first one is of remarkably sophisticated bacteria that is a prokaryotic simplest bacteria, E. coli, but it navigates the space very precisely. It knows where to go and where not. So it can identify its food or toxins very accurately. So how does it do that? It does that because of a cascade of biochemical reactions going on inside the cell. And since this cascade of biochemical reactions, so some reactions happen that switches on other reactions inside the cell, and these finally something comes and binds to the base of the flagellar motor, and overall the cell can change its direction of going. Right? It moves in a run and tumble fashion. So when there is a chemoattractant or they see some food, the run length gets increased and the number of tumbles get decreased. So because of this, it can navigate the space, and for that, interaction of biomolecules, signaling events, and the cell level. So this is an isolated bacteria. So there are no, in this kind of problem, we haven't considered a colony of bacteria. What kind of tools and techniques we can employ here? We can have, there are a lot of inherent stochasticities in the biochemical reactions, so we can use stochastic methods. We can write down master equation, multivariate Fokker-Planck equation, there are time scale separation for reversible, irreversible reactions and different kinds of things happening here. So we can take approximations, we can do Gillespie simulation, which is basically an event-driven process. Since different rates of the things are involved in the process, we can take a kinetic Monte Carlo type of thing here. So now, this bacteria is a rigid cell. While it moves, it cannot change its shape. So now we are going, say we think, oh, okay, one point to remember, what is the speed of this bacteria? Say the speed is 10, on average 10 to 20 micrometer per second in a run in water at room temperature. Right? So when we move to multicellular, now we are not defining it's not a bacterial cell anymore, it can be any cell. So any cell or a cell of a higher organism that actually changes its shape while it moves. So how you can take account of that cell shape or dynamic cell shape morphology into account, you need to invoke different kinds of techniques to have that. So as you see, it can, the example is a kind of a cellular pots model based on statistical physics, and you can do Monte Carlo type of simulation here. Why that works? This operates at a particular time scale and length scales, right? So basically, these are non-equilibrium systems, but still it works because you at that time scale, you can consider the system to be in kind of an equilibrium. It wants to migrate and find its food. Right? There, in the third kind of scenario, we'll take again we have a bunch of cells, but then the cells are not moving. This is another example of where we have cells. Now we have something there is inside, so that's a third example, where we have something going on at the biomolecular level, in terms of genetic regulation. So here in the third model, we connect the 3 levels of this architecture, where you have something going on inside the cell. There is cell-to-cell interactions in terms of, that's a basic interaction in terms of diffusion. Diffusion in free space, we know there are cell membranes and all that, so there are a lot of hindrances, but to the first approximation, we start with the minimal model and have diffusive interactions. So we can invoke reaction diffusion equations, and then we can look at spatiotemporal patterning at the tissue level, and that's a candidate for different kinds of features like self-fed decision-making, disease propagation, and many other things. So all of these these cases have experimental relevance. And so this is a field where you have connections between theory and experiment. Some things from theory we can predict, and some things and highly motivated by experiments, and also some things we can predict from theory. The experimentalist can do the experiments. Sometimes we have a one-to-one mapping, sometimes we don't, but they give very useful insights. So I'll stop here with the spatiotemporal patterning of tissue level, and Vijay will take over, like collective behavior of interacting systems, very nascent field of active matter. Vijay.
Thanks, Ashishi. Yeah, one thing I'll do different is I'll show a lot of movies, okay? Yeah, so the main message is, I mean, if you ask me, what is the most exciting thing that I've seen in the in the recent times, and that is the physics of living matter, okay? And it's also called, like the framework is called active matter, and it is still in the nascent stages, okay? So if you look at, like in our curriculum, we know how to understand the physics of small scales, the physics of large scales, system with many particles, interacting systems. We have a framework for describing this. But the major question here is, can we also describe living systems? That's the an important question. Like, that's one of the thing that has attracted a lot of attention. There is a lot of rapid development in this field. Okay, so we were showing these movies. I'll just try to describe, like, what these movies are. This is a cell in a C. elegans that is undergoing cell division. Okay, so you can see that there are forces that are being generated. There are flows that are being generated. The question is, what causes? Like, what is the reason, and can we model them? Okay, so that is one example that is motivating us to look at the physics of living matter. And the second example is that of birds. They form these flocks. So when you have small number of birds, I mean, they fly in random direction. But when you have a large collection of them, you start seeing that they make these structures. So it's like the question here is, can we propose something like a Ising model? So you all know Ising model. At very high temperatures, you have a paramagnetic phase. If you go to low temperatures, you have a ferromagnetic phase, okay? Disorder to order transition. So here also we are seeing something similar, disorder to order transition. So can we model this phenomena using a Ising-like model, okay? So that is the question. I mean, these are the kind of things that are motivating us, okay, to do experiments. And okay, so let me go to the next. Yeah, so what do we do in our labs? I mean, we look at active matter. Once again, like, the thing that we are looking at is phase transitions in active matter. I'll just show this movie. So in this movie, what you're seeing is these are colloidal particles, and then it's a mixture of colloidal particles, bacteria, and then a solvent, okay? You don't see the bacteria because they're very small. The reason these particles move is because they receive kicks from the bacteria. And what you see here is, like, we start with a disordered phase, and then, like, the system orders, okay? So how does this ordering come into, why does the system order? And how is it different, like, similar, you can see similar kind of phenomena even in equilibrium systems, disorder to order, ordering phenomena. And how is this different from the equilibrium scenario is what we are trying to understand. And we have very interesting results on this topic. Pragya has a poster. We have done experiments, and then we have tried to explain all the experimental results using a theoretical model, and then we'll be soon submitting it to a very nice journal. Please go ahead and have a look at the poster of Pragya. Very interesting physics we have there. Okay, so this is one aspect of phase transition that we're looking at in active systems. The second thing is, once again, like, this is something that my cosmology colleagues would be interested in. Here, once again, like, what we're looking at is active crystals. Like, we start with very disordered configuration, but as the system anneals, you can see that the defects, what you're seeing on the screen, these are defects, okay? 5-fold defects and then 7-fold defects. One can also assign a topological charges to these defects, and then you see as the system anneals, the defects annihilate, and then they also form these string-like structures. So I'd be very happy to discuss with you guys, I mean, at a later point of time. So this is other aspect that Shine is looking at. Probably he doesn't, I mean, he's not presenting, he's presenting a poster on a different aspect. But I mean, yeah, so this is something that we're looking at. And the other thing that we are studying in our lab is how disordered solids behave to mechanical stresses, you know. So it's a very mundane thing that we are trying to understand. You take a solid, you apply stress, it deforms, and then if you apply more stress, it fails, okay? So we all think that, you know, it's a very mundane thing, we understand it very well, but there are a lot of surprises, you know, like, you know, we don't even know how to I mean, what we have tried to do here is, like, you know, we have tried to define defects in disordered systems and then try to show how these materials deform. Okay, that is what is being shown on the screen. Last movie is, like, you know, once again, this is an optical okay, you don't, I mean, probably you can see, like, you know, what Rati has done is, like, you know, she has generated an optical vortex and then trying to deform these 2D monolayers of colloidal particles. And then by doing these kind of experiments, we can understand a lot about how these disordered materials behave to forces or external stresses and all these experiments. And thanks to Pawan, like, you know, he's been helping us a lot in setting up these optical vortex experiments. Yeah, with that, yeah. Thank you a lot.
Perfect. So I have about now 35 minutes, which is actually what we were nudging towards have an elaborate discussion. I would want to first start with kind of, you know, basic discussion of the fact that this kind of spans over molecular scale to all the way to tissues through structures which may not have life, but they can also combine life with non-living objects. So one of the motivations, mainly you can see that it is mainly coming from biological viewpoint. It is a very vast area, complex area, but also there are very interesting physics questions. And that is the reason why we have anchored this particular panel in a physics conclave, because there are also questions which are motivated from a physics viewpoint. So I would want to just get a gist from all of you. What are the physics viewpoints which are available and to be addressed in your own work, and how do you go about addressing those questions? So we'll start with Apratim in the same order what we started with the thing, so that we'll be able to cover. Yeah.
Yeah. So in my particular case, I was doing different kinds of self-organization, how molecules come together and form different structures. Now, since I started to explain, somebody gave me the problem of chromosome organization. Can you have a look at it? Explain what happens. We stumbled upon these so-called entropic interactions. Entropic interactions have been there in soft matter. I mean, yeah. But using entropy to organize segments of polymers. Polymers are intrinsically disordered objects. Who talks about organization in a polymer? I mean, that itself is a contradiction in some sense. Yeah? So getting polymers to organize themselves, that too by putting loops inside it, and those loops are not my hypothesis or creation. Loops exist, except that I design them in a certain way, I tune entropic interaction in a certain way that things get ordered, and I can match it with experiments, multiple experiments. So the polymers with designer loops is something which I would never have looked at. So I'm not only trying to find a biological answer to some observed phenomena, I stumbled into new physics. Topologically modified polymers. I later learned that topologically modified, the term existed, but designer loops, tuning entropic interactions is something which I am currently doing. And I still find it surprising, even as I do it, things are working out the way it is. Polymers with designer loops is something which I would have never thought of just from the physics context. So that's a very concrete example. And I'm sure as we look at more, unknown phenomena, we are not going to solve physics problems. We're going to discover, just solve biological mechanisms. We are going to discover new physics. At least I have done it for my case, right. And I don't know what else.
Yeah. So, Shiva, you have your point.
Yeah. So I think we as a physicist, when you start doing biological physics or physics of life, it's choppy waters. Okay. And when I first wrote a grant proposal to Wellcome Trust DBT, and I was the only physicist who proposed a biomedical question and a kind of a solution to it. I was not also very clear about how to go about doing this. And the audiences, conferences and staff which hardly understand anything. As Pratim said, we left biology in class 12 or whatever. But then the technique that we were using in the lab, which was widely, and there were exciting new papers coming out in biology using AFM and optical tweezers. And what we did just before what we're doing now was the physics of nano-confined water, and we sort of settled the controversy that existed for a couple of decades. And then the next thing for us to do, since that was over, the next thing for us to do, what to do next. And then we sort of walked into this trap of, colleagues keep saying, yes, our colleagues keep saying, are you doing biology, are you doing physics, and so on and so forth. And the question is, why is it relevant to physics? Biology is the question. So when we talk about molecules like polymers and proteins and DNA and membranes, all of these self-organizations happen at the level of energy kT. So if you see a folded globular protein, the energy required to fold it and set it, that tight, nice configuration is of the order of just little above kT. And this means that thermal fluctuations are going to disturb it and, do a lot of things. Now, on experimental front, we do precision measurement, just like our atomic and molecular physics guys do. We measure picometer of displacements. We measure piconewtons of forces. So all these precision measurements have reached a stage where we walked past the thermal limit, essentially. So we are able to measure things which are on its own will now sort of go up and down by more than picometer. And then when you pull on a protein or you press on a protein and try to measure its mechanics, this is quite important thing because you're thermally limited, and what you measure, it seems like broad distribution of forces and how to make sense of it. And then what we are realizing now is that the statistical physics, particularly the one that is of the stochastic thermodynamics, is now is addressing these questions. A lot of theories in stochastic thermodynamics are now built upon the experimental protocols to measure, the bond between two proteins, to unravel a DNA, cause a melting transition. Is melting, but it's not really thermally induced, but force induced. And all of these questions are, are core statistical physics questions. For instance, Jarzynski equality. That's one of the most important example is been proposed. And optical tweezers, Bustamante's lab, has worked on DNA hairpins to show that small degrees of freedom, if you apply forces like this, and if you are able to measure them accurately, he actually established Jarzynski equality in lab. And I think that's where it's not just biological question now. It's a question that is
beyond the domain of
gone beyond. It helps biology, and more than biology, I will say it helps biomedical science. It's like a couple of examples I took from my work, and so I'm at least now collaborating directly with doctors and people from NIV. And I think that is quite fulfilling. And along with that, we are physicists. We still are interested in physics. We're still approaching the whole problem from physics point of view. It's just that more complex.
So Ushishi, this ek tir mein do shikar kind of a viewpoint, it's very effective because the system is already present, but it's complex. And you could see that you're also looking at bacterial system, E. coli especially. It's a system also, I think, our colleagues in active matter also use extensively. Anchoring the physics question in a broader biological problem. Because now if you look at even APS has now a biological physics division.
And also it has PRX Life.
PRX Life, okay. There's a journal which is dedicated to that. There's a lot of push within the physics community towards this. And I can see that you are also working at that particular forefront. Give us a very brief gist of the physics viewpoint of this kind of questions.
I think we can go back and this is a very interdisciplinary institute, right? And we should not actually demarcate physics and biology so much and think about natural sciences, right? And these are the tools and techniques from nonlinear dynamics, statistical physics, complex systems being applied to understand these kind of systems, right? Where you have inherent, where you have interactions and inherent stochasticities involved. So you need physics techniques That's for and there are a lot of condensed matter physicists and worldwide who has come and being thinking of these biological physics, these type of questions altogether, and there are review articles on that title says, "Are biological systems poised at criticality?" Right. Yeah. So these are the titles, and also I would say, so another very important question. So when you look at tissue and you look at spatiotemporal patterning, right? So, and then Apratim is looking at DNA organization. So many students or in many times they have asked, so in a cell, there is metabolic network, genetic networks, and so many things happening side by side, right? All everything is happening. So why are you looking at only these things? Right. So why when tissue spatiotemporal patterning is happening at the tissue level, DNA is organized within the nucleus. These two things happening simultaneously. But you have a time scale separation and then you have to frame the right question and you need to know what kind of things you would like to know and frame questions accordingly, and then choose your techniques, modify your techniques, whatever available, and then address that particular point. Right?
Wonderful, wonderful.
And there are a lot of experimental, there is a plethora of experimental techniques, and there is this feedback between experiments and theories. So there are predictions from theory, and also there is no one-to-one mapping always, but beautiful insights, and you also get to validate the experimental things what you see. Yeah. Right? Yeah. So I think it's a beautiful field to explore and all.
Yeah. So we'll come to Vijay. In fact, I'll take the cue from one of the very interesting things which Aparna was also mentioning related to the more is different. Yeah, the famous Anderson's essay, which actually has become a guiding principle for a lot of emergent processes. And I know that your lab has been looking at even glass physics and other kind of questions from beautiful colloidal physics experiments, including the recent results which have come out from your lab. What are the basic, fundamental, and also very deep questions in physics which you and your group are trying to address in this program?
Yeah, as I said, this whole area of physics of living matter that has come into picture, only in the last couple of decades. So you can imagine the number of open questions that are present here, the challenges that are present here. It's unlimited. Okay, so what we are doing in our lab is just scratching the surface of it, So the grand challenge would be in a few years, one should be able to write Kardar statistical physics 1 and 2, particles and then fields, So active particles and then active field theory. So that is a kind of thing one can do in this area of research. It's so brand new. There's so many things. And these experiments, I have to tell you, we did not really plan these experiments. We were trying to do a very complex experiment of measuring viscosity of bacterial suspension in Noah Khan's lab when I joined here, and it did not go as planned. And then we thought we'll look at very simple problems, and even simple problems are so interesting. So interesting. I mean, if you look at this particular problem that Pragya solved, it's amazing. We were able to look at the theoretical aspects, and then, so once again, if you want to understand this problem, you can go to Kardar 2, Chapter 1, Problem 1, okay? So that explains, the problem of phase separation in equilibrium systems, okay? So now, what we have done is, we have taken that problem and we have compared, what is happening in that, in our system, and then, showed, how this non-equilibrium phase transition in these systems is different from equilibrium phase transition. Okay, very basic question. Yeah?
Beautiful, beautiful. Yeah. So one of the interesting elements is, although the motivation, as you can see, my colleagues on the other side of this particular seating take kind of very interesting problems from biology and also try to address the physics questions. I have to also mention there is another different dimension to this whole problem. Case in point is, let's say, if you take liquid crystals, which actually are one of the most fundamental soft matter systems. In fact, Digenes, who ended up getting a Nobel Prize in 1992, did an outstanding work of bringing theoretical physics ideas right from superconductivity into the realm of some very fascinating aspects of topology-related issues in soft matter system, which still happens to be of very high relevance. So much so that if you now take, let's say, a droplet made of liquid crystals, they actually can show pneumatic defects. And these defects actually can be utilized for various different operations. For example, if you introduce light into such kind of a system, any light, for example, a Gaussian beam, which actually has a Gaussian profile, which is emerging out of this particular topological droplet, can now be converted into a non-Gaussian, topologically kind of active light system, which means that you can encode information, and that is one of the very fascinating aspects of all these systems, what you have also been mentioning. There is also a deep connection to information, which is something, an untapped potential in all these things, because it's also a complex system. There's also a lot of information which is embedded in this. This information can be transferred from one particular pathway to the other pathway, and hence you will be able to do the processing in a very efficient way. So much so that devices have been already produced. Your mobile phones actually have liquid crystal screens. In fact, but for the fact that they are active in terms of the electric field what you apply, you would not be able to actually carry out very interesting changes. And this is all in the realm of soft matter. Because the question of why it is called as soft matter is because you can actually have a small perturbation onto the system, and therefore you would be able to alter the state of that system without having to have a large budget of energy. And that is a very fascinating phenomena. And the other segue I want to now take into the discussion is this element of jugalbandi between the various different aspects of ways of doing physics, right from hardcore theoretical physics with very beautiful ideas into the computational regime where you can also utilize a lot of resources in terms of nowadays even AI and machine learning, and of course experiments. And those experiments actually are relatively kind of inexpensive, relatively, I have to also mention that. And could you also give us that gist, the kind of energy scales associated with that, the time scales, the spatial scales, because all these problems are associated with that.
Absolutely.
So Apratim, you can start and then we can go into the same.
So there are two things I would like to make. So here, why is the KBT so important? As Shiva said, he's measured these molecules, he's giving piconewtons of force. What is the size of a molecule? Nanometers. How much would it move, or if its conformation is going to change?
Biomacromolecule, right?
Biomacromolecule, protein, yeah. So 5 nanometer, 10 nanometer is the size, right? When it gives the force of piconewtons, moves it by a nanometer, if you multiply, the energy scale that you get is KBT. Yeah? So here you need weak interactions because you want the system to be flexible. The other thing, I've been talking about non-equilibrium physics. Non-equilibrium physics has been going on for quite some time. Why suddenly physics of life now? Yeah? So, I mean, you should just see the number of institutes, degree programs, Max Planck institutes which are coming up. So basically, I mean, previous century was of quantum mechanics. When I go to conferences, I hear that this century is going to be about living matter, and I think not 2 books of Kardar, 8 volumes will be required. But all that we are doing is nonlinear dynamics, statistical physics. So here, what Vijay does is non-equilibrium physics, but here the energy forcing, the driving, is coming at a very local scale. Typically, non-equilibrium physics driving is at the boundaries, what was considered or what was being studied. Here, at each degree of freedom, there is some energy input. Now, this kind of system has been done for last 10, 15, 20 years by Vijay, and now they're applying this new physics which has developed where energy forcing is happening at the very small degrees of freedom. That is being what is used in living systems. You have ATP which is coming in locally at different parts of the cell and driving things. Yeah, so unless that stat phase was developed, it wouldn't have.
Perfect.
Okay. And experiments also. Maybe, yeah. But you can talk about
Shiva can actually mention that. So.
Just before we came here, Mukul, I think, at the lunch table, jokingly said that, these are softies. I mean, this in physics, we always call, soft matter guys. It's and for some reason. And the reason I feel, in my opinion, is that most of the condensed matter physics, hard condensed matter physics is hard, not because it's hard mechanically, but it's hard because you do precision measurements. You cool down things, you go all the way to, as Ramki said, microkelvins even, and this is required, and this is amazing feat, technological feat that one is able to achieve. But what we are faced with, that if we try pushing our limits, measurement limits all the way down, we hit at one time, thermal fluctuations. There is nothing you can do about it in all biological systems, in soft matter systems. We have to work at room temperature, and this we can't help. And but let me say, as a counter to what we being called as soft, the limits at which we are making measurements now are optical tweezers, magnetic tweezers, atomic force microscope, and many other techniques. Even in core biology measurements, such as current that is passing through the membrane by opening an ion channel, runs into picoamperes. So these are hard measurements. These are not, in terms of precision, they're as precise as conventional condensed matter physics. In terms of energy, Katie, he already said, in terms of time scales, a protein relaxes from one conformation to another, sometimes in microseconds, sometimes milliseconds, and there are movements that are happening at the scale of nanoseconds. So all of these produce what they call in protein as a microstates, various microstates, and so each time scale is important. But the most significant time scale, which is quite relevant from biological point of view, is the time it requires to take protein to change from one shape to another, because it delivers, like hemoglobin holds on to oxygen, goes somewhere, releases it. These conformation changes are stochastically happening. There's no ATP hydrolysis also in this whole process. It's just thermal kicks that allows this. Yeah. And I guess this is what that makes it interesting also. And in terms of precision, we are as precise as anyone else in physics.
Very nicely put. So we'll take quick comments from the ends. And then,
so one of the interesting aspects that I see here is the collaboration between experiments and theory. It's been great, actually. So there have been a lot of advances in terms of colloidal science, and also techniques coming from biochemistry, and then chemistry, biology. It's a very interdisciplinary field. And there is input.
And I should mention, there's also a lot of debate. On the WhatsApp group, these two guys actually keep asking a lot of questions.
No. So I mean, he's physics of life, and I would like to call it as physics of living matter. The reason is life is just too large. I mean, I would like to live in a small corner where I can identify myself. Life is like a sea. I get lost in that. So that's the only argument. But otherwise, so one of the good things is this. I mean, all agree, there's no disagreement, So so this synergy between the theorist and then the experimentalist, I mean, there are developments happening at rapid pace on experimental side and also on theoretical side. So this is very good for the field. It's going at a very rapid pace, and
Excellent.
But I mean, even though it's going at a rapid pace, the number of open questions are just unlimited. You can just imagine. I mean, just imagine, you're talking about life, what all questions that you can answer. I mean, I just showed you example of cell division. I mean, that is the fundamental process in living, for living, okay? So how does that happen?
We'll move to Ushashi and then take questions.
I'll just say one line. So since you asked about E. coli, so there is this, so you can imagine a micron-sized organism moving in a meter-sized kind of bath, right? So how it will compare whether there is some diffusion gradient there, like whether there is some chemical gradient. So that actually can remember what it has sensed 4 seconds back. And that kind of memory comes because of the biochemical reactions in the real system. And theoretically, we need to put in some memory kernel. There are a lot of things going on in the theoretical modeling, and also there is integral feedback between these systems. So a simple single-cell prokaryotic organism can do these kind of stuffs. Right. So how things become complicated, and also you take different techniques which is mostly physics based, and you need to answer that, where you need to understand about these systems, what I reiterated and emphasized before also. Right. So I think we'll open up for audience questions.
All living creatures ask questions. So the session is open to questions. So if
my question is directed towards Professor
Where is
Usashi.
the question coming from?
My question is directed towards Professor Usashi. You mentioned self-aid decision making. So I just had question that in certain developmental processes, like let's say embryogenesis, it's to achieve the same body plan across, let's say, different individuals. This pattern formation is maybe just not enough. You want the same pattern to form in every individual, and those times cell decides which fate to take, like which distinct type of cell decision it will take, and proportioning is also important. So I understand that we can use statistical physics to understand its self-organizing properties, but how is the field trying to address this reproducibility across all the different individuals where the same pattern is being formed?
Yeah, so there will be interplay of different kinds of tech. Like you would like to know it's like how we can do it from theoretical insights, right?
Yeah, at tissue level.
Yeah, so one thing is that you can apply nonlinear dynamics where you will have some transient time scales and steady state, right? Usually these kind of systems relaxes at long time scales, and long time scale behavior, it is that particular cell fate, right? It has reached to a certain cell fate. Transient time scale is that you are deciding where to go. Right? So there will be a lot of fluctuations at short time scales, and how you look at short time scale behavior and long time scale behavior. So say you are interested in that only the cell fate behavior, then you can write down deterministic differential equation. Like you can write down differential equation, like deterministic part, and not think about the stochasticities. Right? And there you need to think about like what's happening at the shorter time scales and all. So techniques will be different. And also these stochasticities are basically for like many times for modeling, we take these stochasticities into account for like initial configuration, like any cell will have different kind of random initial conditions, right? That starts from, we think that is the, like say the model to start with to account for these kind of scenarios. And they will go probabilistically to different kind of cell fates.
Yeah.
Thank you.
There is one question.
Yeah, so my question is towards Professor Pawan and Professor Chikati. So as far as I understand, your work relates some non-living component like electromagnetic waves or colloidal particles with some living matter. So I don't think it's trying to directly answer any questions about the physics of life, but does it have any application, say in biomedical stuff like nanorobots sending into the blood or maybe surgery or something like that?
Yeah, I'll answer in parts. First, I'll just address this. I actually don't work anything on a biological system. In fact, I'm only the soft part of this thing. My motivation to address questions mainly come from looking at what happens to light once they actually get through the soft matter. And for example, light, if you look at, let's say, a simple equation, a plane wave, if you now consider a plane wave, the omega, the t, the k, the x, and there's also an e cap, which actually is associated with, let's say, the directionality, the k vector. These are all very important parameters when you have to actually process them. Okay, light happens to be an excellent carrier of information, as you probably would be knowing. One of the important questions to address is how do you utilize these particular parameters and encode information and process them. One of the way to also encode information is to impart that particular momentum of the photon, or in this particular case waves, onto the object of interest and look at what happens to these objects. Now, by looking at the mechanical degrees of freedom, you would be able to learn a lot in this particular interaction, point number one. The second point is that once you have set this object into motion, some form of motion, either it be rotation, or it can be even revolution, or it can be a translation, these degrees of freedom can become parameters to process optical information. Okay, and this is also an area called as statistical optics. Nowadays, it's also called as statistical photonics, where essentially you take for example, the degrees of freedom which arises out of the fluctuation of polarization of the light because of the fluctuation in this soft matter system. Now, the question you ask is how do you study the fluctuation? That's where the entropy comes into picture. There's a von Neumann entropy associated with this. You actually can compute this von Neumann entropy from a very precise measurement. And it's usually called as Stokes vector measurement. I usually teach that to our students, in fact, in the optics class. And there are actually 3 important vectors from which you can associate this degree of freedom. So you can encode information, and this has already been used. In fact, the quantum information frontier is frontier, but there is also a lot of optical information, which is not only a frontier, which is actually the multi-billion-dollar industry which is running your internet, which is running a lot of stuff. Now there are new tools which are required to do that more efficiently. And the benchmark, for example, all the quantum information processing people are trying to reach is actually set by the optical information processing. So therefore, there is a lot of importance already associated with that. And therefore, if one can breach through some other methods, well and good. Good luck to them. And that's actually one of the very fascinating problems. So I work only on the soft matter aspect.
Let me answer that question, like how we trying to understand living systems by doing these kind of experiments. Yeah, let me take this example. So here it's the fluctuations. I mean, these experiments are done in a bacterial bath, in a bath that contains bacteria. And you can imagine that the fluctuations of this bath is very different. We call it as an active bath. It's not a thermal bath. The fluctuations in thermal bath are going to be different from the fluctuations in an active bath. And all that we are seeing here is the presence of this, like how these active fluctuations different from passive fluctuations, Brownian fluctuations, or thermal fluctuations. So in a way, like we are trying to understand what kind of fluctuations can you expect in living systems. Yeah, I mean, whatever unconventional features of phase transition that we are seeing here, it's a consequence of the kind of fluctuations we have in the system. So yeah, in a way, what we are trying to understand is indeed the fluctuations due to living systems. Okay,
he has.
Let me complete. So the second example is active crystals. Okay, so these are non-equilibrium crystals. I mean, if you take your solid state physics, whatever you learn is equilibrium crystal. Okay, you look at this Debye behavior, and also you look at dispersion relation. So are they valid for these kind of systems? Because these are out of equilibrium crystals. Okay, so once again, all these fluctuations are related to the living systems, okay? So they cannot, I mean, you don't expect them to see in thermal systems. I hope that answers your question.
Also, like any practical applications using these systems?
Yeah, so once again, we are trying to see whether one can make metamaterials out of these things, okay? So phononic metamaterials, photonic metamaterials. So that's again application side of active matter, which I have not spoken here.
So metamaterial is the individual components don't play a critical role vis-a-vis compared to, let's say, the collective property. So how you arrange plays a critical role. And these are systems where you can arrange very efficiently because you have control over them. That's the advantage. Yeah, Apratim had something to add, and there was a question.
So basically, I wanted to say the these are more control systems. The statistical physics, which we learn, I will learn from him, or I'm learned from him, or his kind. I'm going to apply it within the cell later, once we understand. So these are much more controlled systems compared to that. So I will use it definitely.
Yeah, if I have one more minute. So, this experiment is also very interesting because if you look inside what happens inside a cell, so sometimes you find these structures, they are called membrane-less organelles, okay? So these are just protein condensates, okay? So, people are trying to understand how these protein condensates have different physiological functions, okay? If they stop functioning, you'll have some disease, okay? So, that kind of importance. So, how do these protein condensates form inside a cell? And the emerging picture here is, they form due to phase separation, okay? And then, it's like what we are doing is like a toy model, okay? Yeah. So the next thing that we want to do is we want to put a chain of colloidal particles and then see what kind of phase behavior we see.
Yeah. The student first. No, older student later. One second. Yeah.
So continuing on the previous question, it was said that for organization, like when we have tissues, which forms as a result of divisions, it's not always that the cell remains the same. It undergoes differentiation. So if we use the same differential equations to model what was at the beginning of some time t0 versus when it has already been differentiated and it forms the tissue level organization. So do we like change whatever the interaction is in between, or like do we try to model it as overall?
Yeah, maybe a question for Shashi because.
We take cell division and cell differentiation. These two are two different processes. So we take these two in different umbrellas, right? And these happen at different time scales also. So, and also like statistical physics, so Vijay was we were talking yesterday and Vijay had a paper. So they had so we usually tell that these things work at, so you have to keep the total number of entities conserved in these systems. So cell division happens, right? So somewhere also like in a real system, like there are programmed cell death, which are called apoptosis. So some of the cells at some different positions, so you have a confluent continuous tissue at some different positions, some cells are dying because of some reasons, and some cells are getting divided at different positions. So as a whole, in the system, the number of cells are conserved. So at that limit, we can apply the theory.
Now we'll just take maybe two. Yeah, there is one. Quickly ask. Ask.
We have been seeing we are distinguishing living system from non-living system, but other than these definitions that living system, we see that they can move, they respond to stimuli. Other than this definition, are they been changing over time? Because we can see that non-living systems can now be acting like a living system. So let's say machine learning through AI. So these definitions we need to be clearing up, or
now these questions, for example, the external energy budget has to be accounted for. The living system actually utilize that energy, which is kind of inherent into their system. So in active systems, you have to have, let's say, ATP or some kind of an external source of energy to propel them, do things. So there is a clear differentiation between them. But once you go to smaller and small scale, where do molecules become, let's say, a living organism? Yeah. Is a question for a full-fledged conference.
Yeah, so may I ask one question?
Yeah.
So I'm curious whether the active matter can be trained into a behavior. In a sense, there was one question about memory which she commented. So suppose you have a cell and through a repeated behavior, passing, let's say, through a small microchannel or something, they acquire certain electrostatic and other interactions. And can you have an assembly like that trained into a certain behavior? Because we are self-learn. Is it possible?
There's a lot of motivation for these problems.
I don't know about cells, but for sure there is a lot being done in the area of robotics.
Yeah.
Bots, you can have swarmulators, bots that Santanam was also talking about. So there, you can, program them the way you want.
Yeah, because finally it is a kind of cell wall and everything is like an electrostatic thing and so on. So a repeated behavior, whether it will embed some memory into a cellular system, is a curious question, I think.
So I want to add that there is a field called synthetic biology where actually people entrain these cells and they behave as logic circuits. So they can behave in a certain way, and these are synthetic systems. Actually, people work a lot, and these kind of things happen. And there are maybe there was a discussion of nanorobots and all. So these things, so applications goes to medicine, and also, but that was inherent. So natural system has evolved so nicely that people started to understand what fundamental level E. coli does that. Right? That was a starting point, so that whether we can mimic how E. coli navigates space so precisely, let's say that we need to apply it for targeted drug delivery or designing nanorobots. Right? That was the point.
Yeah, soft robotics is a very big issue. In fact, that's something which is very fascinating. Yeah,
thank you.
Last
Sorry, already some of the questions were already answered. So, just curiosity, as you said here, the equations or the interaction, it is very active or something. So, first question, is there a equation that describes that interaction nicely? And second question, can I set up a millicon drop experiment, which I drop something and see what happens in a fluid? gets a terminal velocity. Here, something like that happens. Like there's a viscosity, I suppose, also over here, right?
Yeah, in fact, these experiments actually, some of them are running in Vijay's lab. Some of them actually are addressing these kind of questions, the viscosity related issues and other questions.
It has thrown up a whole new field of research. You can ask all those simple questions that you had answered, all those first year, very fundamental, very simple questions you can ask now in the context of active matter. And I mean, for sure, these were some of the experiments that I was thinking, and it's just a matter of setting up the visualization.
Two fundamental equations to, for example, Langevin dynamics, where you essentially have mx double dot gamma x dot plus specific external force and a stochastic noise term. And then you actually can introduce more and more complexities into this equation. And it's a very fascinating equation. You can write it down, but the solutions to this equation actually is the trajectory of the particle, where you look at the so-called mean square displacement. A lot of us actually look at that particular part of it. It's a Fokker-Planck equation. That is another one where probably.
So that is what has been done in this paper exactly. You have these passive particles that are being kicked by the bacteria. So how do you model that? So the bacteria gives rise to a noise which is not Gaussian in nature. It has a memory, spatial memory, and also a temporal memory. You can write down the equation of motion using that kind of a noise, and then you see that it reproduces these results, experimental results, in a very nice way.
I see. Thank you.
Just last question, Pawan.
Last question. Yeah. Okay.
So, so Apratim said this KBT energy scale. So I think it would be interesting if there is a energy spectrum, study of energy spectrum around KBT. And probably life has chosen certain stable processes which are above KBT. So which are robust against noises, and the dynamical process which are below KBT. So you have any such comments on that?
May I? Yeah, just. So I think this spectrum is already there, and Robert Phillips' paper in Physics Today, it clearly sets the scale of energy for various processes. And so if you take energy required to fracture a material, let's say if energy required, electrostatic energy between particles, or all kinds of energies, chemical energies, and all of these cross each other at KT at the scale of nanometers, at the length scale of nanometers. And that's where the conversion of energy from one to another becomes extremely efficient in nanoscale systems. One reason why proteins are 4 nanometer object. See, if it was 10 nanometer or 100 nanometer or more or so, then this won't be able to convert energy from chemical to mechanical or from one from to another extremely low efficiency process like our car is today. So if you look at the energy scales, the typical one, 4 piconewton nanometer is the one that we draw a line and see various processes, what kind of forces are involved and what kind of displacement these forces produce. Covalent bond energy is 1,000 times more typically than 4 piconewton nanometer. Ionic bonds are even stronger. So what we're talking about here is the self-assembly or assembly of molecules that have come together just by around KT by being nanometer sized, and that essentially is how sort of life seems to organize itself.
Yeah, we'll actually conclude the session. Thanks to our colleagues who were part of this.